the computer reaches the age too old. The reaction so slow. Okay, so I think the change of the title is much easier to to pronounce it. Yeah. Okay, so this is my second talk. So in this talk, I will mainly, to repeat the what I did the first talk, to give the more detailed information about the type 1 APP signaling pathway in neurogenesis, and also this long title for the AICD, how the AICD is, through the MER63, transcriptionally modulated the neurogenesis. So yes, so the Claude just mentioned, in some AD patient, so the A beta deposition, hardly to find it. But what's exactly the function of the APP, in the AD still is quite a puzzle. So our group is mainly, as I mentioned before, mainly focus on the intracellular part of the APP, the AICD, after cleavage with the gamma secretase. So we know the notch pathway, so you see like the DERT1, and also in our group we also find the F3, which can interaction with notch, and also this interaction, extracellular interaction, can trigger the notch intracellular part release. So they had many the function include the oligodendrocyte differentiation. And similar to the notch, APP also is the transmembrane protein. It's the type 1 transmembrane protein, and the APP can also degrade by the gamma secretase. But for many years, nobody knows. So which ligand, the extracellular ligand can, play the role, the interaction with the APP, which can trigger the gamma secretaries, the cleavage and the release of the AICD. So our group, work on this topic for many years. So here is the first the experiments we did. You see, here is the CHO transfected the type 1 cell, and here is the CHO transfected APP cell. We coded the APP FC under the dish, and here we coded the type 1 the FC under the dish. Then so played this cell, type 1 cell, and the APP cell to the APP and the type 1. We can see the cell can, the APP type 1 cell can bind into the APP, and the APP transfect cell can bind into the type 1. And this interaction can be blocked by the type 1 antibody, and also can be blocked by the APP antibody. So definitely we have the more experiment, to confirm the interaction between the APP and the type 1. So further we try to understand, so whether this interaction, type 1 APP interaction, can release the AICD, can be triggered the gamma secretaries, the activity, to cleavage the APP and release the intracellular part of the APP AICD. So here is the result. We treated we can find that we use the type 1, can treat the CHO cell, CHO APP cell. So it's hardly to find this. So I don't know. So this is the problem. So okay. So here we can look. So the we can find that after trigger extracellularly triggered with the type 1 FC, the APP CHO transfector cell can release the more it's dose dependent release of the AICD in the intracellularly, and we also checked the AICD concentration in the type 1 Y-type mice, the heterozygous type 1, and also the type 1 knockout mice. Oh my God! Here also we can find in the type 1 knockout mice, the AICD release is reduced, significantly reduced. So the further we try to understand whether the interaction type 1 the APP interaction, after this interaction, the AICD released, whether this AICD has the function, the transcriptional function. So we use the Thomas-Sudolph system. This is the luciferase assay system to test the transcription activity of this type 1 APP interaction. Here is the result. You can see the type 1, we confirm the type 1 regulate AICD-dependent transcription activity in the CHO cell. Here is the CHO cell. This is the CHO L1 transfect cell as the control. Here is the CHO tax. It's the human type of the type 1. And here is the CHO type 1 transfect cell. So after we transfection with the full length APP, the GAL4, the full length the version, so into this cell, we can find the transcription activity, the AICD transcription activity, significant increase in the tags and the type 1 cell. But if we transfect with the truncate, only the intracellular part, no cannot interaction with the extracellular part of the molecule. So this form, you can see the no significant increase. And here we can also further confirm, if we use the gamma secretase inhibitor, which can reverse the AICD transcription activity. So this is the repeated further confirm the type 1 APP interaction can activate the gamma secretase activation. So yeah, this is the summary. So the type 1 APP interaction can trigger the gamma secretase activity and release the AICD. So our next question. So after the AICD release, what's this signaling function? What's the final readout of this AICD? So we try to understand this. We did the double staining, type 1 and the APP in the embryonic neural stem cell niche. We can find the APP can, express in the SVZ region, and the type 1 also express this region, and the APP and the type 1 colocalization in the SVZ region. So the SVZ region we know. So this is the one of the area which can produce generate the new neuron. So we further did the experiments for the neurogenesis experiment. We isolate the neuronal stem cell from the SVZ region. We did the the neurogenesis assay. You can see. So this is the APP Y-type Y-type mice. So this is the APP knockout mice. You can see that in the APP knockout mice. So the tubulin. The staining, you can see is increased, and the MAP2, the staining also increased. So indicate, the neurogenesis in this APP knockout mice is increased. And here we also find that in the type 1 knockout mice, so the 2G1 and the MAP2 is increased, indicate in this knockout mice, the neurogenesis is also the increase. And here we also did the type 1 APP double knockout mice. So we still find that the neurogenesis is a significant increase. So here we also try to to understand whether the AICD type 1 can inhibit neurogenesis in the type 1 knockout mice, but this type 1 is cannot inhibit the neurogenesis in the type 1 APP knockout because they lost the ligand interaction. Here is the result. We can see that in the type 1 knockout, if we play, added the type 1 FC in the different dose, you can see the neurogenesis is reduced. And here in the type 1 APP knockout mice, if we add the type 1 FC, you cannot find the neurogenesis reduced. So this further confirms the APP and the type 1 interaction is essential for this kind of signaling pathway. So here is another confirmation. We transferred the AICD into the type 1 knockout the neuronal stem cell. We can find the AICD can definitely block suppress the neurogenesis. So this is another result to confirm this kind of interaction. So this is we coded with laminin and the F3 as a control, L1 as a control. So in the type 1 APP double knockout, you can see that only the type 1 FC can significantly increase the fold of the transcription activity. And here, this transcription activity can be blocked with the gamma secretase inhibitor. So this also further confirms the AICD, definitely played the role in the transcription, as the transcription factors. So we know that the AICD interactions with FE65. So we also checked the neurogenesis in the FE65 knockout mice. You can see the expression of the FE65 also in the SVZ region in the neural stem cell niche. And here is the neurogenesis the study, the FE65 wild-type mice and the knockout mice. So in the knockout mice, the neurogenesis also significant increase. So this means the FECD5 also works as an associated factor for the AICD. So we did all the experiments we did in the embryonic stem cell. We further asked the question whether the type 1 APP interaction, the pathway also occurred in the adult neurogenesis. So we can find that both molecular type 1 APP also expressed into the adult neural stem cell niche. But this is very hard to look so here. So here is the result. We analyzed the neurogenesis in adult, the type 1 knockout mice and APP knockout mice. You can see the neurogenesis is a significant increase in both type 1 and the APP knockout mice. And we also did the analysis in the similar analysis in the APP type 1 double knockout mice. So this is the adult. We can also find this is a significant increase. This indicates the type 1 APP pathway, the signaling pathway also occurred in the adult neural stem, the adult stage. Here is the result of the FECD5, the adult knockout mice. Also, you can see this FECD5 knockout mice, the neurogenesis significantly increase. So this is the summary what we did in 2008. So the type 1 APP both express the embryonic stem cell, neuronal stem cell, and also adult neuronal stem cell. Type 1 APP is the interaction in the extracellular matrix, which can trigger the AICD release through the gamma secondary the cleavage, and release the AICD together with the FECD65 migrate into the nuclei and surprise this migration surprise the neurogenesis. So after these experiments, last 7 years, my group was further to try to understand how AICD played a role in the surprise of the neurogenesis. As I mentioned before, they had two pathways. One pathway is a direct pathway. Type 1 directly modulated the expression of the target gene. For example, of the P53, the EGFR. So we set up another, the hypothesis. We asked the question whether AICD can through modulate the, the microRNA, then so indirectly, modulate the target gene expression, then so, suppressing the neurogenesis. So we did the AICD chip-seq experiments in 2007. Actually, that time this technique just developed in Singapore. We transfected the AICD into the neuroblastoma cell, the human neuroblastoma cell. We used the antibody, to recognize the AICD. We pull down the AICD binding domain in the chromosome. We did the full sequence of this, the AICD binding domain. So we repeated this experiment twice. You see that we can find that this 2 times the experiment they has about 70 or 80% the overlap. So they had about 280 microRNA promoter region. So the repeat can be repeated in these two experiments. We also confirm, so through these experiments to further confirm the, the liability of this, the AICD chip-seq the result. We also found the AICD can bind into the promoter of the P53 and also the promoter of the EGFR. So further confirm this experiment is reliable. So the first analysis, we pick up the the top 20 from each experiments. So they almost had the 12 microRNA is overlapped. We further analyzed this expression of the 12 microRNA in the neuroblastoma cell, trigger with the AICD. So we can confirm, certainly, about the 8 microRNA, so definitely, overexpression after the AICD, the regulation. To further confirm this, we also, use the human neuronal stem cell, to check the expression. Finally, we screen about the 4 microRNA, which can also expressed in the human neuronal stem cell, which is under the regulation of the AICD. To further here we also to identify the exact binding site of the AICD in the chromosome. So you can see this is the 4 microRNA which regulated by the AICD. They definitely have the binding site, the significant binding site located in the chromosome. So this is also the first time to confirm AICD played a role as the transcription factor which can bind into the region of the certain microRNA, promoter region of the certain microRNA. So for the function part, we, transfection this 4 microRNA into the neuronal stem cell. We did the neural stem cell neurogenesis assay. Finally we found the MER663 can definitely, downregulate the neurogenesis. This is the result, from the facts, the analysis, and this from just the counting the result. This is the, and we further confirmed this, whether, the co-transfection, the AICD and the inhibitor, the MER663, into the neuronal stem cell, we can find that the inhibitor can reverse the AICD inhibitory effect. You can see here. This is the facts result. This is the counting result. So from this 4 microRNA, we identified that only 1 microRNA played the role in the surprise of the neurogenesis. So next part, we ask what's the specific the gene modulated by this MER663. So we did the 2 time point the microarray analysis. So we transfected the MER663 and the AICD into the neuronal stem cell at the different time point. It's the 12 hour and 24 hour. So from this we can find from the 12 hours we find the 7, and from from 12 hours we find the 7 genes overlap, and here we find about the 11 gene overlap. Then we further, to check the expression of this gene, 18 gene in the neuronal stem cell. So we further, to confirm this gene, whether this gene is associated with the neurogenesis. From this 18 gene, we find the 4 gene, involved in the neurogenesis. It's never have reported this. So this gene. So this gene and this one is 4 gene. So let me summarize this almost 15 years work. So so we found the tag 1, the interaction with the APP in the extracellular part, and this interaction can trigger the AICD release. So after release, AICD migrate into the nuclei, and this migrate the AICD not only modulate the certain gene expression, for example, P53, EGFR, but also can modulate certain microRNA expression. And this, for example, the MER663, through this pathway, the MER663 can also modulate some targeted gene which involve in the neurogenesis. And so we have no idea about this, newly identified the 4 gene, how this gene, play the role in the AD, or maybe the AICD, the APP, and also Tau's, pathology. So this is maybe our next step. We want to continue to work on this. So let me thank my the students and the collaborators, so many collaborators. So, okay, thank you so much. Any question?